This is DNA in its classic double helix form, and it's from X-ray crystallography, so it's an accurate model of DNA. If we unwind the double helix and unzip the two strands, you see these things that look like teeth. Those are the letters of genetic code, the 25,000 genes you've got written in your DNA. This is what they typically talk about, the genetic code. This is what they're talking about. But I want to talk about a different aspect of DNA science, and that is the physical nature of DNA. And it's these two strands that run in opposite directions for reasons I can't go into right now, but they physically run in opposite directions, which creates a number of complications for your living cells, as you're about to see, most particularly when DNA is being copied. And so what I'm about to show you is an accurate representation of the actual DNA replication machine that's occurring right now inside your body, at least 2002 biology. So DNA is entering the production line from the left-hand side, and it hits this collection, this miniature biochemical machines that are pulling apart the DNA strand and making an exact copy. So DNA comes in, it hits this blue donut-shaped structure, and it's ripped apart into its two strands. One strand can be copied directly, and it can be seen spooling off down to the bottom there. But things aren't so simple for the other strand because it must be copied backwards. So it's thrown out repeatedly in these loops and copied one section at a time, creating two new DNA molecules. Now, you have billions of this machine right now whirring away inside you, copying your DNA with exquisite fidelity. It's an accurate representation, and it's pretty much at the correct speed for what it's occurring inside you. But I've left out error correction and a bunch of other things. This was work from a number of years ago. Thank you. This is work from a number of years ago, but what I want to show you next is updated science. It's updated technology. So again, we begin with DNA, and it's jiggling and wiggling there because of the surrounding supermolecules, which I've stripped away so you can see something. DNA is about 2 nanometers across, which is really quite tiny. But in each one of your cells, each strand of DNA is about 30 to 40 million nanometers long. So to keep the DNA organized, to regulate access to the genetic code, it's wrapped around these purple proteins. I've labeled them purple here. It's packaged up and bundled up. All of this field of view is a single strand of DNA. This huge package of DNA is called a chromosome, and we'll come back to chromosomes in a minute. We're pulling out, we're zooming out through a nuclear pore, which is sort of the gateway to this compartment that holds all the DNA called the nucleus. All of this field of view is about a semester's worth of biology, and I've got 7 minutes, so we're not going to be able to do that today. No, I'm being told no. This is the way a living cell looks down a light microscope, and it's been filmed under time-lapse, which is why you can see it moving. The nuclear envelope breaks down. These sausage-shaped things are the chromosomes, and we'll focus on them. They go through this very striking motion that is focused on these little red spots. When the cell feels it's ready to go, it rips apart the chromosome. One set of DNA goes to one side, the other side gets the other set of DNA, identical copies of DNA, and then the cell splits down the middle. And again, you have billions of cells undergoing this process right now inside of you. Now we're going to rewind and just focus on the chromosomes and look at its structure and describe it. So again, here we are at that equator moment. The chromosomes line up, and if we isolate just one chromosome, we're going to pull it out and have a look at its structure. So this is one of the biggest molecular structures that you have, at least as far as we've discovered so far, inside of us. So this is a single chromosome, and you have 2 strands of DNA in each chromosome. One is bundled up into one sausage, the other strand is bundled up into the other sausage. These things that look like whiskers that are sticking out from either side are the dynamic scaffolding of the cell. They're called microtubules, but the name's not so important. But what we're going to focus on is this red region. I've labeled it red here, and it's the interface between the dynamic scaffolding and the chromosomes. It is obviously central to the movement of the chromosomes, but we have no idea really, as to how it's achieving that movement. We've been studying this thing called the kinetochore for over 100 years with intense study, and we're still just beginning to discover what it's all about. It is made up of about 200 different types of proteins, thousands of proteins in total. It is a signal broadcasting system. It broadcasts through chemical signals, telling the rest of the cell when it's ready, when it feels that everything is aligned and ready to go for the separation of the chromosomes. It is able to couple onto the growing and shrinking microtubules. It's transiently, it's involved with the growing of the microtubules, and it's able to transiently couple onto them. It's also a tension sensing system. It's able to feel when the cell is ready, when the chromosome is correctly positioned. It's turning green here because it feels that everything is just right. And you'll see that there's one little last bit that's still remaining red. And it's walked away down the microtubules. That is the signal broadcasting system sending out the stop signal, and it's walked away. I mean, it's that mechanical. It's molecular clockwork. This is how you work at the molecular scale. So with a little bit of molecular eye candy, we've got kinesins, which are the orange ones. They're little molecular courier molecules walking one way. And here are the dynein. They're carrying that broadcasting system, and they've got their long legs so they can step around obstacles and so on. So again, this is all derived accurately from the science. The problem is we can't show it to you any other way. Exploring at the frontier of science, at the frontier of human understanding, is mind-blowing.